On behalf of our ISPE Education Committee, I'm Emily Bratton, Vice Chair of our ISPE Education Committee and Webinar Subcommittee Lead, and I'm Senior Director in Applied AI Science at IQVIA. I'll be moderating today's session. Before we get started, I just want to mention your lines are muted. If you need tech support or have general questions, please use the chat function. If you want to pose a question to our panelists today, submit that question at any time in the Q&A feature in your display bar. And at the end of the session, we'll be coming through those questions and answer them in the order in which they were received. If there's not enough time, we'll try and follow up with you on those responses. I know this is a really important topic to many folks joining the call today. This session is recorded and will be made available through the ISPE website in the coming days. As many of you know, cell and gene therapy products often require extended safety and effectiveness monitoring with follow-up that can go up to 15 years per patient. And while we have clinical trial extensions that can contribute useful evidence, real-world data are especially critical to sustain the necessary duration of that follow-up and capture specific data elements over time. At the same time, real-world data-based long-term follow-up studies really face these distinct challenges, including sample size constraints, capturing data and data quality, that can really be more pronounced than other real-world data studies given the longevity of the follow-up period. The topics we're going to cover today are to enhance our understanding of the regulatory, scientific, and operational requirements of cell and gene long-term follow-up from a global perspective. Our panelists today will provide examples using real-world data and how novel approaches such as decentralized study designs and tokenization can support the capture of crucial data throughout long-term follow-up. And before we dive into this exciting agenda, I would love to introduce our distinguished speakers and my colleagues who bring a wealth of experience and expertise from across the field. Lisette Hugendorn is an epidemiology director at IQVIA in real-world solutions. She has extensive experience in epidemiology and pharmacovigilance with a special focus in regulatory requirements and the use of real-world data in drug development, inclusive of cell and gene therapies. Jamie Geyer is the global head of quantitative safety and epidemiology at Novartis. Her work covers a broad range of therapeutic areas, including gene therapy and neuroscience, which are highly relevant to today's topic. And finally, I'm really pleased to welcome Nick Honig, Executive Director of High Learner Health, and his background spans regulatory leadership at Aetion, legal counsel at the FDA, and strategic roles at Pfizer. He's passionate about advancing real-world evidence and supporting innovative approaches for long-term safety monitoring and gene therapy. So without further ado, I'll turn you over to Lisette.
Thank you, Emily. Thank you for the nice introduction. Good morning, good afternoon, everyone. I would first like to share some further context for the topic of this webinar, and as an employee of IQVIA, what you just saw is my disclaimer. As you can see from the figures on this slide, the field of gene therapy medicines products is growing rapidly. The figure on the left shows the growing number of authorized gene therapies over time in different major geographies. By the end of 2023, in total 29 gene therapies were authorized across these geographies. The US, European Union, and Great Britain had the highest numbers of approved products, and Canada, Japan, Australia, and China had lower numbers. By Q3 of 2025, in total 38 gene therapies had been approved across different geographies. The figures on the right show that over 2,000 gene therapies are in development and that the majority of these are for oncology and rare disease indications. The number of therapies in development that is in the pre-registration phase is still limited, but for phase 2 and phase 3 the numbers are higher and the number of therapies in the preclinical phase is very large. Next slide, please. Are you able? Oh, thank you. Gene therapy is considered part of the group of so-called advanced therapy medicinal products, ATMPs in short, together with cell therapy and tissue engineered products. If we look at the specific characteristics of gene therapies, we see the following. First, compared with small molecule and biological medicines, they are characterized as highly innovative and personalized medicines with distinct mechanisms of action. Gene therapy requires unique methods of manufacturing and administration. Treatment involves the administration of living cells or specific genetic material. Second, the treatment is ultimately targeting modification of gene expression in patient cells, and this requires a vector to insert genetic material in the cell. This can occur in vivo, which means that a direct modification is made in the patient's cell in the body, and the alternative is ex vivo, which means that cells are first harvested, then modified outside of the body before administration of cells to a patient. Traditionally, viral vectors have been used, but novel platforms also make use of non-viral vectors. Third, when we look at the diseases that are considered for treatment with gene therapy, these are ones with specific cellular or genetic aberrations for which no or unsatisfactory treatment exists. Examples are inherited diseases or refractory cancer, and thus diseases with high unmet medical needs. Fourth, looking at marketing authorizations for gene therapies, which are frequently for rare diseases, pivotal evidence is often based on either single-arm trials or very small randomized trials. Given the unmet need, expedited regulatory pathways are applied. Next slide, please. To further illustrate manufacturing and administration of gene therapy, I would like to give an example of a specific type of ex vivo gene therapy, which is referred to as CAR-T cell therapy. As shown in the figure on the left, for this treatment, blood is obtained from the patient and subsequently T cells are separated and removed. These T cells are then genetically altered to have special receptors, which are called chimeric antigen receptors, abbreviated as CAR. These CARs are designed to recognize a specific antigen on cancer cells. The so-called CAR T cells are then grown and multiplied and introduced in the bloodstream of the patient. Different CAR T therapies have been approved for the treatment of specific blood cancers. The CAR T cells identify the cancer cells with target antigens and kill them. Later in this webinar, Jamie and Nick will provide examples of long-term follow-up studies for CAR T therapies. In general, ex vivo gene therapy approaches are used for conditions affecting tissues that are either accessible or easy to get in and out of the body, such as blood and skin. Next slide, please. Let's now move on to what makes it so important to study long-term safety of gene therapies. Given the specific ways of manufacturing and mechanisms of action, there are particular safety concerns that will also be listed in risk management plans for these treatments. These include delayed adverse events of special interest, which may not be seen for many years. First, secondary malignancies. These can be caused by integration of viral vectors that are used to insert genetic material in the cell. Second, immunogenicity can be triggered by long-term expression of the therapeutic protein. Third, persistent or latent infections can occur because some viral or bacterial vectors used can remain dormant and ultimately reactivate, causing infections, especially in immunocompromised patients. Fourth, unforeseen effects from vectors presence or prolonged transgene expression can cause organ damage or other issues, resulting in long-term toxicity. Finally, genome editing-based gene therapy may have off-target effects and thereby increase long-term risks. Next slide, please. This webinar is organized on behalf of the ISPE Cell and Gene Therapy Special Interest Group. And I also would like to share that this special interest group has a relatively short history. It was initiated only in 2022, and at that time we also began working on a paper on long-term follow-up after authorization of gene therapy. This led to publication of an ISPE-endorsed paper in Drug Discovery Today in spring last year. Given the large number of gene therapies currently in development, which I've shown, the knowledge of how long-term follow-up studies of gene therapy can be appropriately designed and operationalized to ensure optimal collection of safety and effectiveness data in routine practice is essential. Next slide, please. I will now go over regulatory requirements for these studies. Gene therapies require extended monitoring due to their novel mechanisms and uncertainty about long-term outcomes. And this need for long-term monitoring starts with patients treated with investigational medicinal products or gene therapies in clinical trials. Most regulatory agencies in different geographies have specific guidance for gene therapy clinical development. Clinical trial participants must be followed for a much longer period after completing the parent trial. At the same time, confirmation of safety and effectiveness in the post-authorization phase is also important, as for other newly approved medicinal products. There are fewer formal regulatory guidances focusing on long-term follow-up of patients treated in routine care after approval of gene therapy. The focus in our paper was on non-interventional studies in the post-authorization phase, but it's important to acknowledge that harmonization or even integration of long-term follow-up of clinical trial participants and patients treated in routine care can be beneficial. Next slide, please. This brings me to the key regulatory guidances that are currently in place that cover concerns regarding delayed adverse events and sustained effectiveness for authorized gene therapies. The EMA first guidance is from 2008 and was revised in 2018, and it focuses on risk management of ATMPs. There's also EMA guidance from 2009, which is more focused on follow-up after administration of gene therapy in particular. The FDA guidance is from 2020 and focused also on follow-up after administration of gene therapy. A more recent FDA draft guidance that came out in 2025 is not particularly focused on the requirements for long-term follow-up, but it rather points out different approaches for data collection that can be used to capture safety and efficacy data post-approval. Next slide, please. As you can see on this slide, the EMA and FDA guidances not only focus on general considerations for post-authorization monitoring, but also provide recommendations for the design of these studies. This includes guidance for the required duration of follow-up, which may very well be up to 15 years. Further, there's guidance on the generation and assessment of preclinical and clinical data to assess risks and safety concerns. There's also attention for considerations regarding specific types of gene therapies. Finally, risk minimization measures and risk evaluation and mitigation strategies are also addressed in these guidances. Next slide, please. The FDA guidance from 2020 also includes a framework with questions that can be asked to assess the risk of gene therapy related delayed adverse events. As you can see from the visualization of this framework on this slide, the answers to the questions help in assessing the need for studies with long-term follow-up. Next slide, please. With this, I'm handing over to Jamie, who will tell more about the role of real-world data in gene therapy long-term follow-up studies.
Thanks, Lisette. Hi, everyone. My name is Jamie Geyer, and what I get to share in the next 10 to 12 minutes makes me so happy because it really was the crux of why the Cell and Gene Therapy SIG was established a few years ago, and I think speaks to the specialness of ISPE and how we can come together when we're tackling methodologic and operational considerations for these studies and trying to develop strategies to answer questions for key stakeholders. And when a group of us came together, we were thinking, who are the stakeholders here? It's our patients and their healthcare providers. But it's also the questions we're receiving and some of the wrestling that healthcare authorities are going through, academics, and then pharma at large. And I know I'm biased, and I know this group is a little biased, but we truly felt when we came together that epidemiologists were best suited to answer these questions, to apply the learnings, to apply our methods, to secure data really to make informed decisions for patients, healthcare providers, and other stakeholders. So go to the next slide, please. This is my disclaimer as an employee of Novartis. We'll go to the next one, please. So registries and fit for purpose. These are the things I say on a regular basis. I don't know how many times everyone here says fit for purpose in an average day, but I go between 2 and 22 times a day, I think. And it's because it always reorients me to my objective or the objective of what we're trying to achieve as scientists. And that is, what is the question we're seeking to answer here? And are there data fit for purpose with the elements that we need to answer those questions? And I've had the distinct pleasure over time to work with many registries, largely disease-based registries that have helped me to do that. Some have common data models that facilitate analyses over geographic regions and other spaces. Some have linkages to other databases, whether it's mortality or oncology. And some lower the burden for patients, particularly when you think of retention-related issues because of the systems in which they work. But I want to bring everybody's attention over to the left side of the slide, because those are some of the elements we wanted to highlight and we focused within the paper that Lisette mentioned earlier, is that, of course, one registry, it's not a one-size-fits-all sort of situation, and that there might be additional data collection that is required, specifically when we're looking at this new area of cell and gene therapy, and that we almost have to put on that hat of the primary data collection studies to ensure that we have everything documented in a protocol and that we're doing things in a standardized way. And when we were going through some of these points, thinking about fit for purpose and what is feasible, we came up with some 10 points to consider that are common to most of the epidemiologic work we do. It makes me smile when I think of all the work that was done in parallel. Whether we're talking about ICHM-14, whether it's the SPIF2 framework and other platforms that have been proffered lately. You look from, defined study population, exposure, covariates, linkage to other data sources. Do the registries have experience with non-interventional studies? But really, making this list is a way for us as scientists to think about what are the sources of bias and confounding that could lead to gaps in our understanding, and what can we do either through study design or analytic methods to adjust for those or to plan for those accordingly. And then what is left behind for all of us from a transparent perspective to showcase where those limitations are when we disseminate this information. Go to the next slide, please. When we were going through this effort, we also did a bit of a landscape analysis of what's available today. Of course, this is not an all-inclusive list. The area is evolving rapidly. These registries were chosen in part because they've been used for long-term follow-up studies for cell and gene therapies, but also they work in spaces in which these therapies are under development. So oncology, rare disease, as an example. In addition to all of the criteria we have listed here across the columns, I think the beauty of these registries is what else they get to offer. And I think often about spinal muscular atrophy, or SMA, which is, depending on the subtype, a certain death sentence for babies. And what we've seen with effective medicines are these babies develop into toddlers, develop into adolescents, and beyond. So with respect to the natural history of disease, these registries are really giving us a glimpse in what does it mean to have SMA today or to be diagnosed as a baby with SMA. There are other elements where they're talking about prognostic factors that these registries give insights to, treatment outcomes, of course, as well as treatment durability. Go to the next slide, please. The representation that we have here, it's just 2 long-term follow-up studies in gene therapies to help concretize some of the points that we were trying to make, not only in the paper, but as people working in the protocols and analysis plans for cells and gene therapy. And that is, the fit for purpose element. So we have the indications listed here: multiple myeloma, TTD, and severe sickle cell disease. The objectives here, very similar for the studies under evaluation: safety as well as effectiveness. Study design, cohort study embedded within a registry setting, the same registries, as you can see here. But the objectives of the study are different. One is a descriptive study, not requiring a comparator group. One was a comparative study, and therefore embedded within the registry setting a comparison by which to make analyses against. Data collection, the one on the left, pure secondary use of data. The one on the right required additional supplemental information, which they worked with the registries to ensure that they could collect that data to do analyses to better understand the long-term safety outcomes of interest. Sample size were different based on the objectives of the studies, but you can see that follow-up duration of what we're talking about before is of up to 15 years. Go to the next slide, please. I'm going to switch over a bit to operational challenges, and you're going to see we've tried to place them in 3 buckets. I'll move from left to right. So you have the study population and patient retention, data collection. Of course, the other side of it that all of us worry about is missingness, and what does that do with respect to our study and the analyses that we can do down the line, and then some treatment-specific elements to keep in mind. A theme that's not obvious in how we've done this, but I want all of us to keep in the back of your mind, is about the challenges, not just to us as researchers, but to the patients and healthcare providers managing these medicines on the day-to-day basis. So let's start with the study population. First, there's the transition of care. We have patients often transferring back to the healthcare center that's local, their community center, from a specialty center. What does that mean in terms of continuity of data collection and the information we need for our studies? Pediatric patients, as I mentioned with SMA, growing into an adulthood, change of healthcare providers is a certainty, but also you don't have the guardians and parents who are kind of the gatekeepers of this. The point of contact changes as well. There have been many solutions that have been proffered, from decentralization approaches that we'll tap into in a bit. But obviously that requires buy-in from healthcare providers and others over a very long period of time, and that we have to work with patients to ensure that they make sense and are feasible. In our middle panel here, the data collection and missing data, I know many of us have worked on studies that have continued for a long period of time, and standard of care changes and should change given the work that we're doing with medicines. But there are data elements that may no longer be meaningful, some that may no longer be collected. What does that mean for us at the end of 15 years when doing analyses? I was part of a study, to the point of the second bullet, in which the primary healthcare provider was a neurologist. And we quickly found out that we needed data from an oncologist, GP, OBGYN, and pediatrician. How do you combine those data over 15 years period of time? It becomes ever more complicated, particularly for the patient, his or herself. And then we have the treatment-specific elements. What if you are given a one-time gene therapy results in cure? Wonderful. But in terms of the research, the interactions with the healthcare system decrease significantly. Causality assessments, delayed adverse events. We've wrestled with this as epidemiologists for a long time, but this is a unique piece. How do you measure this? It's not about switching medications and causality in that way, but rather, do we need more information, tapping back into that primary data collection element? Molecular surveillance, is that required? Do we need tumor tissue samples? And then, of course, is the element of rare disease and dealing with privacy considerations with patients. You go to the next slide, please. When I was talking about the operational aspects, I did highlight the patient in particular and healthcare providers, because I think we all agree it's not just the right thing to do, but it's arguably critical for all the work that we do to inform study feasibility, to answer questions in a robust fashion. And the SIG came together in talking about this and how best to incorporate that voice for benefit risk evaluations, getting perspective on data collection methods, and obviously PROs or PROMs. But some of the things that I've experienced as a researcher recently is patients told us that their experience had been reduced to one symptom, and it wasn't even the most important symptom to them. So how could we have a study that was looking at one outcome as an example? So that required changes to the design, to the questionnaire, and a more holistic approach from our perspective. As part of this SIG, we also had a discussion recently about how do we operationalize endpoints and what's meaningful. If all of us sit back and think, if I say the word mobility, what does that mean to us? Does that mean being able to walk across a room, being able to sit up? Or in some cases, does it mean just the use of a pointer finger to be able to manage a device to make it across the room, for example, a wheelchair? Feasibility, we've talked a lot about this, and burden, distinguishing between what is feasible for pediatric patients and their caregivers as compared with adult patients. We talk about wearables or online platforms and having the voice there to understand what is meaningful and capturable, but also, is there a way for us to align that with standard of care visits so that we're not adding to the problem? And then the patient-reported outcomes, as discussed before, not necessarily a part of standard of care, thus making it important that we put that into our data collection tools and do it in a smart way. And all of this has to occur throughout the study phases where we're talking about the design to evidence generation. But one thing our SIG really wanted to emphasize, and is something that's often missed, we talk about the real physical burden, time burden for patients, but oftentimes we miss the emotional burden, the trauma that's associated with having a serious disease, and or the trauma associated with treatment options. Survival guilt, going back to the clinic in which you're treated and being one of the very few patients still able to do so. The idea that I am well now, it's time to move on. So that's something to keep in mind, because we need to meet people where they are and work together to answer questions for physical as well as mental wellness. Move on. Next slide, please. It's a nice publication. The link is below if anyone needs it. I think the bullets here would resonate with everybody and things that we know in terms of participation in long-term studies, travel-related challenges, and understanding about what these studies are, these long-term follow-up studies. But I urge you just to look at the percentages that are below them. The participants here, it's something we need to keep in the back of our mind to think about from a burden perspective. The last bullet here says most patients and caregivers are comfortable with modifications. Most in this survey, 76% of respondents were comfortable with modifications. And this includes our long-term follow-up studies that would allow direct access to EHR data. Let me go to the next slide, please. And this is my final slide. It's another slide in which I smile, which is why we're all here today doing this webinar, because we're coming together through ISPE to look for opportunities to partner with one another. And I think it's really a beautiful and special place in which we can do so, in which we talk about regulatory guidance, that all of us can feed into this. So when you see something come out for information or feedback, that we do so proactively as United Front. Disease-specific alignment. Let's learn from what people have done to date, particularly as we move into new disease areas and therapies targeting different aspects of a person's life and existence, while keeping the stakeholder in mind at all levels: patient, physician, and health technology assessment organizations. And then with that data source assessment, again, going back to fit for purpose, I'll be my sixth time saying it today, but really looking at what are the optimal design elements, the data sources, acknowledging where there are gaps, owning that, sharing that, and figuring out collectively how to be better. So I now turn it over to Nick, who's going to talk about some novel data collection approaches. Thanks for being here today, everybody.
Thanks, Jamie. Great presentations, Lisette and Jamie. And yeah, I'm Nick Honig. I'm the executive director at Highlander Health. And I am here today to talk about the promise of novel data collection approaches to capture crucial data in the context of cell and gene therapies. So if we go to the next slide, I'd like to start with a little bit of level setting. We affectionately call this diagram the snail, and we use it to talk about how the evidence generation landscape has shifted over the past 25 years. So on the left-hand side, we have sort of the traditional model of evidence generation. You start with a phase one trial, move to phase two, move to phase three. Your drug or medical product gets approved. Sometimes there's a phase four, but there's a lack of continuous data generation, and we see a little bit of a data void in the post-approval space. What we've seen over the past 25 years is a shift. We're moving towards a more continuous evidence generation paradigm. So the right side represents the new reality. We're seeing early approval with better continuous data, as well as some streamlining of the phase one, phase two, phase three model. We see phase one, phase two get combined a lot, and we're seeing the FDA commissioner talk about seamless clinical trials as ways to further improve the efficiency of this process. And this new model really has its roots in oncology and rare diseases, where the nature of the condition really forces innovative approaches. But we really saw it coalesce during COVID-19 when we had to assess mRNA vaccines. These vaccines were approved on a smaller body of information than vaccines usually were and pushed key questions to the post-approval setting, referring to things like duration of effect, booster timing, mixing and matching of different types of vaccines, etc. But now that we're firmly in 2026 and we are moving forward with this earlier approval with better continuous data paradigm, there are a few implications that we need to consider as we think about how to use this data and what we're seeing in the landscape. The first, I think, goes without saying, is that we're seeing an increased demand for data and evidence generation. The second, which follows, is that there's a little bit of a tweak to the definition of how we're viewing totality of evidence for a medical product. Clinical trials are not necessarily enough to carry the day anymore. Clinical trials plus real world data and real world evidence are now considered table stakes to understanding how a drug is performing. The third implication is we're seeing an increased focus on that post-approval or commercial space. And fourth, with all this new data available, what's really interesting about it is that it's now available for personalization in ways that it wasn't before. And if we go to the next slide, I want to key in on that point a little more. So when we're talking about conducting impactful research in 2026, when you think about the story of a person's life, you really need the 360-degree view. And what this slide is meant to illustrate is the multitude of data needed to paint that picture and how we can use the longitudinal timeline as an organizing principle for clinical research. An RCT is a fantastic tool. It will never be replaced, it will never go away, but it's a snapshot of a period in time. Patients are purposefully selected, the medical product is administered in a controlled environment, and there's an attempt to minimize the noise of day-to-day life. But it doesn't give you the full picture. It doesn't let you know how the drug is working in the real world, how it may be interacting with factors that may have been smoothed out during the inclusion-exclusion process. So we also need to think about data collected during episodes of care. Now, this is visits to the doctor, data collected in the EHR, so on and so forth. And some of the things we need to keep in mind when we're assessing this data and this part of the 360-degree view is that this data may not be collected in the routines of a clinical trial. It's almost certainly not. It may not have the formal expectation of cross-checking. It actually may be comprised of entirely different data elements depending on what the doctor is assessing and how the standard of care is changing over time, as Jamie mentioned. And there are other data elements as well, and I'll talk about those in the next slide, and all of these are needed to build that complete and coherent longitudinal story. And if we go to the next slide, one of the most important things to keep in mind as we build better evidence generation systems is not overburdening the current healthcare system. We can't ask more of clinicians and we can't ask more of patients. More formalized check-ins are just not in the cards. We run the risk of toppling the system over. We're already seeing overutilization, overburdening of the ability to provide care. So we need to think about how we can generate impactful data sets that leverage every data element collected in the healthcare ecosystem. It's an imperative for us. So whether it's in a health system, whether it's in a clinical trial, whether you're looking through the EHR, or you're looking through at-home tools like sensors or wearables, or other new data sets that are emerging like genomics data, as we think about this next generation of clinical research, a new set of possibilities arises when we unlock the potential of multimodal data. which is how we refer to sort of all these different data elements in one place. So if we go to the next slide, I want to talk about some of the growing pains with this process and why this process has been so slow. And there are a number of operational aspects that are coming to the surface that we've been doing a good job of tackling as an ecosystem, but there's still some more work to do. As we think about explicitly using data from all these different paradigms in research, we need to think about getting a lot of things right. We need to deal with the data quality mismatch that comes from different data collection approaches in different settings. The data you're collecting in RCT, as I mentioned earlier, is not the same that you're going to be finding in EHR. And specifically, we need to be able to first quantify those differences, and two, have mechanisms to systematically improve it. I'm an attorney by training. I like to joke that I'm reformed because I'm no longer practicing, but I'm always thinking about, from a regulatory perspective, if you're using multimodal data in an application to get a drug approved, how do you demonstrate quality, provenance, traceability, fit for purpose, and all of its glory to regulatory auditors who are assessing this data for its credibility? And as a result, we as a community need to think carefully about things like metadata and guaranteeing sufficient underlying data quality to truly enable data liquidity or the repeated reuse of this data, because that's how we unlock evidence generation at scale, is being able to comfortably and confidently reuse this data for multiple purposes. So if we go to the next slide, we are seeing a lot of progress in this ability to unlock data liquidity. It's a bit of a clumsy metaphor, but we like to sort of compare some of the advances to intracellular calcium channels. So for those of you who may not know, these channels release calcium ions from internal storage sites in the cell into the cytosol, where they act as second messengers to trigger cellular processes, essentially moving information across fixed barriers. In the earlier paradigm that I was discussing, that 25-year ago view of evidence generation, early stage research and development, drug discovery would operate potentially in a silo from late stage clinical development, potentially in a silo from post-approval regulatory data generation and approaches. And we're seeing a blending of this idea and a reformatting of how we're approaching them. So if we think about some of those early use cases and how multimodal data can be deployed, we can think about things like improved target discovery, better toxicology work, indication refinement, and so on and so forth. Moving towards that late stage clinical development area, we're now able to better target label expansion opportunities and improve pricing and reimbursement conversations that we're having. And key to all of this is regulatory authorities becoming much more sophisticated end users of real world data and multimodal data. We're seeing a concerted effort to bringing these data sources in front of reviewers. There's an effort to speed reviews as well as improve surveillance and monitoring capabilities. And some of those proverbial calcium channels that are helping us along are new standards in exchange formats like JSON and FHIR, tools like APIs that allow for bulk data exchange, and improved metadata to help assess quality over the chain of custody of a data element. And all this level setting brings us to the topic of today, which is how these novel data collection and generation approaches can improve long-term follow-up for cell and gene therapies. So if we go to the next slide, a very successful approach to date, as Jamie mentioned, has been the rise of pre-competitive registries. And one of the ways that I like to think about a global pre-competitive registry is almost as deploying a master protocol. Neutral third party, often a patient advocacy group or an academic consortium, creates a single master infrastructure to monitor patients treated with any gene therapy or cell therapy for a specific disease. And two of the biggest success stories to date are the Center for International Blood and Marrow Transplant Research, the CIB-MTR, and the World Federation of Hemophilia Gene Therapy Registry. So starting with the CIB-MTR, this registry partners with biopharma companies to track long-term outcome data. And a recent success story has been their partnership with Kite on a post-marketing safety study for Yescarta. And so this is a prospective post-marketing study for patients with relapsed or refractory large B-cell lymphoma. And I'm classifying it as a success story because it was recently announced that they completed enrollment for the study 2 years ahead of schedule. That's a massive achievement and really shows the power of how these registries can work. We still need to let the study play out, sort of see how that 80% dropout that Jamie mentioned works, but completing enrollment ahead of schedule is a massive, massive achievement. Another notable registry in the space is the World Federation of Hemophilia Gene Therapy Registry. So this is a prospective observational longitudinal registry, and it's set up to determine the long-term safety and efficacy of factor VIII and factor IX gene therapies. It includes a comprehensive data quality assurance program and also collects patient-reported data, which is really important to that multimodal data conversation I had earlier. And perhaps most notably, it's endorsed as a global standard by EMA, which is an incredible achievement and means that a regulatory body has comfort in what that data can be used for. So at Highlander Health, one of our major lines of work focuses on learning labs and demonstration projects that answer pressing questions facing evidence generation. Our goal is to set up these projects and then provide open source guidelines on how to tackle some of the underlying infrastructure of these issues. And we're kicking off a project in the coming weeks that addresses cell and gene therapy long-term follow-up that I'd like to talk about on the next slide. While we were setting up this project and working with our partners at a regional health system, we identified two problem statements. The first is that there's a critical need to decentralize elements of the clinical research project process in order to make it easier for individuals to participate in these multi-decade studies. We need to bring study procedures to the patient instead of bringing the patient to the study procedures. We hypothesize that this will increase the likelihood of long-term study participation and reduce the chance of early withdrawal from these studies. Now, the reliability of collecting necessary data for these studies through decentralized approaches needs to be verified before it can be adopted. I think that goes without saying. So the first goal of our demonstration project is to conduct a cross-sectional cohort study that quantifies the difference between data capture paradigms. We're going to be assessing in-person visits versus hybrid visits versus fully remote visits. So that's the first component of the project. If we go to the next slide, the second problem statement that we identified is that regardless of visit structure or format, collecting necessary data can be overly burdensome or difficult. Aligning with my earlier remarks on the promise of multimodal data and the need to reduce the burden on researchers and caregivers, we hypothesize that the future of these long-term follow-up studies will be an amalgamation of multiple data collection approaches, and they'll be combined to achieve the optimal balance between high-quality data collection while lowering the burden for the patient. And something that we have to keep in mind is that 15 years is a really long time. People get married, people move, people get new jobs, things happen. And to assume that someone is going to take time out of their day to keep coming to a doctor's visit or keep jumping on a Zoom to answer questions is not necessarily realistic. And we need to get imaginative and creative about other ways to collect data, especially with all the data elements that are available for us. And so we fully acknowledge that some data should only be collected in person, but we hypothesize that reliable medical record retrieval, inclusive of additional paraclinical testing data, things like lab tests, imaging, electrophysiology, can provide the primary substrate for long-term safety and effectiveness assessment. So the second goal of our demonstration project is to provide a practical understanding of how automated data collection from a variety of sources in cell and gene therapy long-term follow-up studies should be structured. And if we go to the next slide, we do plan to make all of our findings open source as we do with all of our projects. And as we're looking at what's to come, we plan to create a baseline by using data from in-person visits at the specialty clinic conducted by trained study staff with fully manual curation of medical record into a case report form. The process of proving out the second goal of the project will involve medical record retrieval, tokenization, and linking to paint as full of a picture of the patient as possible, really trying to unlock the promise of multimodal data. And some of the outcomes of interest we're going to be looking at, first, comparing the rates of missing data between visit paradigms. So what are we seeing versus from in-person, virtual, or hybrid? How can we ease that burden for patients and providers? Second is comparing the rates of variance among patient and clinician-derived data. Third is quantifying the impact of strategies for reducing the data collection burden for remote visits and community-based clinician visits. And then finally, getting to that second goal that I mentioned, demonstrating that tokenizing recruited patients and linking them to mortality data, claims data, and conducting medical record retrieval is a viable strategy to access clinical records and capture needed variables for the time horizon we're looking at over the long term. I know that that was a lot of information. I know that we have a Q&A that we're getting to, but I want to turn it back over to Lisette to talk through a few conclusions before we jump into the discussion.
Thank you, Nick. Yeah, and indeed, to summarize, we would like to share some conclusions, mainly also going over what have we discussed today. So first, we hope that this webinar has clarified why long-term follow-up is crucial for assessing gene therapy safety and effectiveness over time, and that we're really talking about very long-term follow-up for these treatments. Second, we have shown that there are multiple examples now of how incorporating real-world data from different sources can enhance an understanding of gene therapy outcomes in diverse populations. However, addressing different operational challenges is vital for effective long-term follow-up studies. Novel data collection approaches, as Nick also explained, facilitated by technological advancements may further support successful long-term data collection, and increased opportunities for data access and interoperability may also play a role here. But finally, we also need to acknowledge that the crucial point is that to make the very long-term studies a success, it's crucial to engage patients effectively, to ensure that we focus on meaningful outcomes, also from their perspective, and also that the required long-term commitment to participate in studies is kind of assured, because we will need them to make that possible, even if we have the perfect technology. Next slide, please. So, and finally, I also would like to acknowledge all contributors to the ISPE endorsed paper of the Cell and Gene Therapy SIG. They are listed with their names and affiliations on this slide. And yeah, I also would like to echo Jamie in saying that I've found it a really nice experience to be part of this special interest group. So we also welcome new joiners. I mean, it's a really nice forum to discuss things, different aspects that are important in this field. And with that, I would like to hand back to Emily for the Q&A. Thank you.
Thank you, everyone, for joining this terrific presentation, and my gratitude extended to the presenters, Lisette, Jamie, and Nick. We'll now transition to an open Q&A session for the remainder of the hour. So if you have a question, please use the Q&A feature. I've been pulling questions over, so we'll go in order. Because your lines are muted, we wanted to reduce the noise. I'll go ahead and read your question. And again, if not fully addressed or addressed today, I'm happy to sort of take those offline and provide them back to the panelists for further follow-up. So our first question that came in is about the elaboration on the process to introduce supplemental data collection to existing registries, specifically for long-term follow-up of treatment evaluation. And if any of you have any sort of key lessons you've learned in those collaborations with registries on augmenting that data collection.
I can start if that's okay. This might seem a little touchy-feely, but it's been quite positive, to be honest. So the people that are running the registries and entering these data and collecting these data over time are doing it for a reason, and it's to get answers to questions that are plaguing all of us. And I found that, honestly, sitting down at a table and discussing the needs, what the objectives are, what health authorities are asking, what your company is asking, what your client is asking, and explaining that to the registry owner opens up communication immediately. There are some logistics. I was part of a study where we actually had to reconsent everyone to provide a linkage to a database that was critical for the study to enter the database. But now I've seen a trend with other databases, particularly in the cell and gene therapy space, which is amazing, that on their landing page, they say they have the capabilities to do so, recognizing the novel aspects of the medication and the questions that are being asked. And that obviously taps into the bit about the long-term follow-up that's required. So I think there's an openness and understanding that when addressed from the beginning permits feeding in and adding additional questions to questionnaires and the like.
Yeah.
Mm, Jamie, I think you hit on a key point there. I know that it was a sort of an experience that you had, particularly around reconsenting patients, but I think that I'm not the first one by any means to have this thought, but I think that the current model of 100% consent on day one and then having to go back and reconsent each patient is no longer viable, especially as these therapies target broader and broader indications. If you think about things like sickle cell. And I think that if I had the answer on how to fix informed consent, I'd be in a very different position. But we needed to start thinking about making it a dynamic relationship between patients and researchers.
Yeah. What I maybe would like to add, I agree with Jamie that often people are very willing, but because they see the purpose of all this, so they're very supportive. But I think depending on the type of organization that is behind the registry and the size, there can be sometimes they're positive, but maybe without really knowing what they're up to and what will be expected from them. Also from a regulatory perspective, when we look at safety data collection, for example, there are quite specific requirements, and that requires expertise also actually kind of within the registry to be able to meet expectations. So I think there's also a big role for all the stakeholders involved to make sure that we can educate registries and also support them to make it possible, because a lot of these things do require a big investment.
I agree. And understanding that as, the evolution of the implementation of treatments and the change of natural history, the information and data collection at the core of the protocol that started may be shifting over time and sort of advising and guiding registries even on their core data elements in addition to supplemental information that they might be collecting for a specific therapy. There's a lot of questions around retention and loss to follow-up and sort of the ability to demonstrate best efforts to the agencies. Number one, experiences with that and engaging with the authorities, but then also how to handle that methodologically, and anything that you might have learned over time. Again, knowing that many of these haven't reached the 15-year mark yet because these are novel therapies, but what sort of analytic methods have been, sort of accepted as far as accommodating that missing data or loss to follow-up?
I think that are a lot of questions, or a lot of aspects in relation to missing data. So maybe I can make a start. I think a key thing is, of course, we cannot avoid loss to follow-up, but I think the key part is how can we stimulate that also for patients. It's clear that it's also a benefit for them to continue in these studies. And I kind of always thought that it seems we have different things in terms of pharmacovigilance. We have additional pharmacovigilance activities, these studies, and we have risk minimizations to explain the patients that there could be certain risks they need to be aware of. But I also think it can be very helpful to maybe as part of that risk minimization to more or less, connected to also show to the patient that you want to educate them, but also that it's clear that there's still a lot to be learned and that they with their data can contribute to that. And in that sense, I found it interesting to see that one of the examples that Jamie shared of a long-term study for treatment in sickle cell disease, among others, that there in the SMPC, in the warnings and precaution sections, it's explicitly called out that patients are expected to be in long-term follow-up studies. And I think that and also have a proper discussion with patients and engaging them in the setup of the study can hopefully also kind of enhance the participation throughout these years. But at the same time, we do acknowledge that we will have missing data. And I think there is still a lot to be learned because some of the studies have only recently started, of course. So I think the true 15-year follow-up we have not yet achieved. But maybe Jamie can also comment on that further.
Yeah. Methodologically, I really can't because we're all struggling with the same thing and what does it mean and what's the bias that's introduced. But I can speak to the other piece is that I do believe that health authorities and those of us doing studies are kind of in it together and learning from each other. And while the expectation is 15 years, I actually attended a webinar a few months ago where a regulator was speaking and said, you know, if I had my choice, I would follow these patients for the rest of their lives. So trying to put yourself in that mentality, if we want to know the most that we can, with recognition that that's not possible and working together to say, we've done our best. This is what we have every year, whatever that interim report is. What do you think? This is the feed. This is what we're learning along the way. I loved the information that Nick was sharing, or doing studies to explore these different collection techniques to demonstrate with data what is feasible. So we're all kind of in it together, working back and forth. And I don't think, at least from my experience, that health authorities have said, you're doing this wrong, you need to do more of this. It is, wow, we are seeing the drop off as expected. What more can we do together? What do we put in the guidance? What sort of data do we need to see to demonstrate effort? What sort of patient engagement activities are underway to make this easier? Technology is exploding, and I just think we're in an amazing space to do more and to bring that to epidemiology overall. So it's, not concrete answer. Sorry, I know that's not always satisfying, but I've found regulators to be extremely receptive and honestly kind of in it with us.
And in a sense, getting the endorsement that in 15 years from now, the technological capabilities and the healthcare data that we're able to amalgamate on a single patient's journey will ultimately change. And
Exactly.
so my experience has been that, the as far as best effort, documenting the strategies that have been used to both recruit and retain patients over longitudinal follow-up is exceedingly important, inclusive of perhaps reconsenting when they hit adult age, because many of these programs start in the pediatric population, especially for gene therapy, and also thinking about it's oftentimes a small numbers challenge, so that loss to follow-up is quite crucial. But ultimately, the analytic methods from the beginning may have been more descriptive to start. So understanding if there is a censored or loss to follow-up subject, is there any sort of information we can gather? Did they decline to continue participation? What were the reasons why? Being able to understand the missingness or perhaps the gaps in care if a patient, say, is lost to follow-up for a period, but maybe we can find them in secondary data down the road. Nick, there's a question for you, and I'm happy to sort of elaborate as well, but can you provide a little bit more information on tokenization for global studies and not just US focused? And perhaps the expected attrition when linking fragmented data sources with that tool?
Yeah, absolutely. I think it's important to preface my answer by saying I'm not an epidemiologist or an outcomes researcher, so I'm not going to speak to necessarily to the technical aspects. And Emily, I'd love for you to weigh in there. I
Maybe the
think
next webinar.
I think that tokenization on a global scale is difficult for many reasons. I think it's difficult enough in the US with our fragmented healthcare system. But when we run into regulatory barriers, things like GDPR in the EU and all the different restrictions on how health data can be used, as well as the standardization issue of getting like-to-like data elements to make sure that you're following the patient and measuring the same things. I think it's difficult. I think these pre-competitive registries help a lot, but I think that there's a lot of work to be done, and we really need to push towards regulatory convergence rather than harmonization. That's something that my friends at the Duke Margolis Institute for Health Policy like to stress is that it's time for us to converge on a set of standards rather than trying to align definitions and really push in the same direction. And I think that there's a lot more work to be done there, but we're making progress.
Yeah, and there is no one size fits all, as Jamie Jamie put. So to use your words as well, Jamie, I am smiling. This was a great conversation. I really want to thank you again for your presentations in today's webinar. As a reminder, this is recorded. It'll be available on the ISPE website in the next few days, and an email will be sent out with the link. Keep an eye on your email for this information about this and future upcoming webinars. We'd love your continued participation and engagement, and we hope to see you next time. Again, thank you all for joining, and we look forward to furthering this topic offline and the collaborations, hopefully, that will come from it. So thank you all.